Hello everyone, warm greetings to you all. Today I'm going to explain one of the important basic topics in the course of animal physiology: signal transduction through neuromuscular junction. In the higher vertebrate animals, the nervous system is highly evolved and also has a complicated signaling mechanism. There are three kinds of neurons are involving for the signal reciprocation. They are sensory neurons, interneurons, and motor neurons. A specialized sense organs senses the chemical and mechanical stimulations sent to the central nervous system through the sensory neurons. Central nervous system interprets the stimulation with already existing memory and send the signals to the target organs through the motor neurons. This coordinated action makes the organism to reciprocate according to the signals given from the external environment. Neurons usually transmits impulses as action potential. So one neuron sends the action potential to another neuron. This series of transfer of action potential lead to transmit the signals from different parts to the central nervous system and vice versa. But importantly, the skeletal muscle has to act. Only then the stimulation can be fulfilled. Nerve impulses, also known as action potential, travels from the brain or from the spinal cord to trigger the contraction of the skeletal muscles. An action potential propagates down the motor neurons to the skeletal muscle fibers. The site where motor neurons excites the skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the point of contact between axon terminals of the motor neurons and the end plate of the skeletal muscle fibers. The event of the signal transduction occurs in 7 coordinated steps. Step 1. An action potential travels the length of the motor neurons to the axon terminals. Step 2. Voltage-gated calcium channels open the calcium ions diffused into the axon terminals. Step 3. Because of the binding of calcium ions with the synaptic vesicles releases acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the gap between the axon terminals and motor end plate called synaptic cleft, bind with the receptor which contains ligand-gated cation channels. Step 5. The binding of acetylcholine opens the ligand-gated channels. Step 6. Sodium ion, represented here in red, present in the extracellular matrix, enters into the muscle fibers, and the potassium ions, represented here in blue, exits the muscle fibers. The greater inward flux of sodium ions is relative to outward flux of potassium ions, causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches the threshold value, an action potential propagates along the muscle fibers called sarcolemma. So neural transmission to the muscle fiber ceases once the acetylcholine is removed from the receptors. Due to that, ligand-gated channels are closed. The removal occurs in 2 ways. 1. Acetylcholine diffuses out from the synapse. Number 2. Acetylcholine is broken down by the enzyme called acetylcholine esterase that breaks the acetylcholine into acetic acid and choline. Choline is then transported to axon terminals for the resynthesis of acetylcholine and construct the synaptic vesicle to receive the signals. That's all about the signal transduction through the neuromuscular junction. This is Jeremiah, Department of Zoology, Bishop College. Thanks for watching. Have a nice time.